Hello students, welcome to EPG Pathshala. I am Mohan Chand Joshi, Assistant Professor from Jamia Millia Islamia. Today we are going to talk on a module, Protein Expression Vector, under the paper, Genetic Engineering and Recombinant DNA Technology, Part 1. After completing this module, you will be able to understand what are the features of a protein expression vector, what are the properties of a expression vector, and how a recombinant protein is expressed and purified via expression system. Protein expression vector are usually plasmids of bacterial origin, and plasmids in bacteria are the extra chromosomal copy of DNA. Plasmid harbors genes which are transcribed and translated independent of chromosomal DNA. Plasmids often improve genetic fitness. For example, in case of antibiotic resistance genes, lot of antibiotic resistant genes are present on plasmid. So a bacteria can get an antibiotic resistance if it acquires a plasmid into the cell. Therefore, a susceptible or antibiotic susceptible bacteria can become antibiotic resistant and can grow in presence of an antibiotics if it can acquire a plasmid containing antibiotic resistance gene. As I mentioned earlier, most of the plasmids most of the expression vector are a plasmid of bacterial origin. They have specific properties which are tabled in this, which are mentioned in this table. So there are, on the left side you have elements or the characteristics, and their description is given on the right hand. The expression vector has origin of replication, which allows the duplication of that plasmid. It contains multiple cloning sites, which allows the cloning of gene of interest in that area. It also contains promoter. It contains selection marker, which is usually in antibiotics resistance genes, which is required for the screening of recombinant cells. Also has a short-peptide tag, which can be attached on the N-terminal or the C-terminal of a recombinant protein, and a primer binding site. This could present, this stretch of DNA can be used by primers, where primer can bind, and this is usually used to confirm whether a gene of interest is cloned in the vector also in not, and also to confirm whether the frame, the sequencing, the cloning is done in frame or not. Origin of replication. So in plasmid, ORI is the reason where the duplication process for a plasmid is initiated, because it allows recruitment of initiation factors. In plasmid, replication is regulated by two proteins, ROP and ROM. But this initiation process and the regulation of this replication initiation at plasmid is different from what we see in a chromosome locus. So in minimal ORI region in ORI in chromosomal DNA is regulated by DNA-A CK protein in combination. In expression vector, they are present in multiple copies. So the genomic copy is a single copy of DNA, while these plasmid exist in multiple copy, and this results because the ORI is mutated in plasmid, which allows the multiple copies of these plasmid to be inside the cell. But advantage of these multiple copies is that you have more protein production in the cell. Multiple cloning sites. Multiple cloning sites are another characteristic of protein expression vector, which are short stretch of DNA that contain multiple restriction cut site, up to 20 sometimes. And these restriction enzyme sites at MCS should be unique and should not be present in the vector backbone. That means the restriction enzyme sites which are going to be used for cloning purposes should not be present in the vector backbone, because in case we want to insert our gene of interest with specific restriction enzyme site that is present in the vector backbone, and we digest our vector to linearize it, instead of getting a one fragment, we will get 2 or 3 fragments depending upon number of restriction enzyme sites present in the vector. The advantage of multiple cloning sites is that if you have across vectors, it allows cloning, subcloning of our gene of interest. So we can move one gene from our cloning vector to expression vector because of these multiple cloning site. Here is an example of restriction enzyme site present at the multiple cloning site or MCS of PET28A, and as you can see, in that stretch of DNA from 1 to 153 nucleotide, we got multiple restriction enzyme site concentrated in such a short region of DNA. However, not all of these sites are unique, so researchers often use those restriction enzyme sites that are unique in the MCS for the cloning purposes. Another characteristic of expression plasmid is presence of a promoter. In compared to a cloning vector, promoter is often missing in cloning vector because we don't need to transcribe. All we need to do is to propagate and increase the copy number or increase the concentration of our plasmid. On the other end, expression plasmid are dedicated for expressing the protein, therefore they contain promoter. And a promoter is defined as a DNA sequence that is responsible for initiation of transcription for our gene of interest. A promoter sequence often contains a combination of DNA sequence that allows recruitment of RNA polymerase as well as operator sequence. In this schematic, as you can see, the promoter region is highlighted here, and we have -35, -10 region as well as the transcription initiation site or start site. The entire region is where RNA polymerase binds, and then +1 site represent where the transcription or the mRNA is being transcribed. Here are the example of bacterial promoters that are being used in commercially available protein expression vectors. There is range of them. Most of them or all of them are of phage origin. So for example, in this case, T7 promoter, which is isolated from T7 bacteriophage, is used for in vitro transcription experiments and also for constitutive protein expression. The requirement for this promoter to function or to produce a transcript requires the presence of T7 RNA polymerase, which should be present in the host cells. And in this case, the transcription is constitutive. That means there is no regulation. If you clone your gene into this in a vector where there is a T7 promoter and you have a T7 RNA polymerase, the expression or the transcription of that gene will be constitutive. T7 lac promoter, which is a fusion of T7 and a lac operator system, is again T7 is from bacteriophage origin. This particular promoter is used for high level expression of plasmid and present in a lot of commercially available expression vectors. This particular promoter allow a very tight regulation of gene expression, and only in presence of IPTG or inducer, this promoter will allow the recruitment of T7 RNA polymerase as well as expression of our gene. Similarly, from T5 bacteriophage, we have T5 promoter. Again, another variant of it is available as T5 lac, which is a fusion of T5 bacteriophage and the lac operator. Again, these T5 and T5 lac can be constitutive or can be tightly regulated as T7 lac. Another one is sp6, is isolated from sp6 bacteriophage. Again, it can be constitutive if we provide sp6 RNA polymerase, and this also been used for in vitro transcription assays and for protein expression experiments. Continuing to our list of bacterial promoter, TRP1 is another promoter that has been used, and as you can see is a tryptophan operon based. As you can see it is used for high level expression of protein. This is constitutive in absence of tryptophan, but can be repressed if we have high concentration of tryptophan. So this is a promoter which can be regulated. Another one is Arabad. Again, it is a promoter from arabinose gene. It allows a constitutive expression, and in presence of arabinose, which removes the repressor, will allow a constitutive expression of our gene of interest. Similarly, there is lac operator, which is lac promoter, which is the promoter from lac operon gene, and it allows protein expression, and it can be induced by IPTG or by lactose. Another promoter is PTAC, which allows protein expression. It is a hybrid promoter between lac and tryptophan, and this is regulated by lac promoter and can be induced by IPTG. Another one is PL. This is promoter from lambda bacteriophage. It allows high expression of proteins, and its expression is regulated by temperature. Another property that protein expression vector has is a short peptide tags. So these are stretch of DNA which encodes for small peptide tags, and they can be at the 3' end or the 5' end of the multiple cloning site, and can be tagged to our recombinant protein or to our gene of interest at the N terminal or the C terminal. Tagging of these short peptides allows two major functions. They can be used for recombinant protein purification, which is a downstream process by using a suitable matrix, or can be used to increase the solubility of the protein, as well as to increase the stability of protein. Further, these tags, once the protein is purified, can be removed by proteolysis, and these can be used also for cellular localization of the protein inside the cell, or to pull down protein DNA complex or protein-protein complexes. Short peptide tag. Here is the list of tags which are available commercially with different expression plasmid vectors. So in this table we have tag, what is the length, what are the metrics that is used to capture these ones, and what are the elutants that are used to remove these tags from the matrix, and what are the applications. The very first one is albumin binding protein. As its name suggests, it binds with the matrix is albumin based, and it can be eluted by denaturations using heat or urea. This is used for purification, increase expression and solubility. So as I told you earlier also, short peptides can also be used not only for purification but also for protein folding and increasing its solubility. The other one is alkaline phosphatase. This is bound, the matrix is made up of monoclonal antibodies, and the protein or the tag can be removed with a buffer which has lower pH. This is specifically used for detection and purification purposes. Another one is calmodulin binding protein. These are, again, the matrix is made up of calmodulin, AGTA or AGTA, and it can be eluted by buffer containing high salt, used for purification and expression purposes again, and then chloramphenicol acetyl transferase. These tags, the matrix is chloramphenicol. It is used for detection and purification as well as increased solubility. Another one is cellulose binding protein. The matrix is cellulose, and it is removed or eluted by denaturation, and it is used for purification, secretion and immobilization. Another one is a chitin binding protein, which binds to chitin. The matrix is of course chitin, and it can be eluted in presence of thiol containing reducing agents such as DTT, beta-mercaptoethanol, and this one is used specifically for purification purposes. In continuing with our list of short peptides, the very first one is a flag epitope, which is a very short tag. It's 8 in length, 8 amino acid in length, and the matrix is monoclonal antibody, and it can be eluted in presence of low pH buffer, EDTA or flag peptide. It's used in detection and purification. And it is noteworthy to mention here that flag epitopes are used for cellular localization as well as for chip analysis, where we pull down protein DNA complexes as well as to pull down protein complexes as well. So flag has been extensively used. The next one is glutathione S transferase. The matrix is glutathione, and in reduced conditions or reduced glutathione, this is used for purification, increased expression, solubility or immobilization of protein. The next is maltose binding protein. It's a very large tag, and the matrix is of cross-linked amylose. Detection, purification and increased expression of the protein. Another one is a MIG tag. It's another tag which is a small peptide, 11 amino acid only, and the matrix is made up of monoclonal antibody, and it is eluted in presence of low pH buffer. It's used for detection and purification. Similar to flag tag, MIG tag can also be used for cellular localization, pull down of protein DNA complex or protein-protein complex, and it has been extensively used in bacteria as well as in eukaryotic system. Interestingly, NUS-A is another is a transcription factor in bacteria. It's also been used for protein as a short peptide tag for increasing the protein expression as well as the solubility. It's a large peptide tag. The matrix and elution is not yet been characterized or been available commercially, but this can be used to increase expression level. Another one is a polyarginine tag, which can be 5 to 6 amino acid. The matrix is cation exchange resin, and in presence of a high pH salt gradient, proteins can be eluted. And in the last table of our short peptide tags, the tag we will discuss about is polycystine tag. It's not that commonly used, but it's available. Its length is 4 amino acid. The matrix is made up of thiopropyl cephalosethiol, and it can be eluted in presence of reducing agents. It's primarily used for purification purposes. The next one is very commonly used and available extensively in PET series of vector is polyhistidine tag, and this tag is available in different other expression vector series also. It's very commonly used. The matrix is made up of divalent metal and can be such as nickel NTA, and it can be eluted in presence of imidazole or at a low or in presence of buffer with low pH. It has been used for detection, purification and immobilization. Similar to flag and MIC tag, polyhistidine tag or HIS tag can also be used for pull down assays as well as for cellular localization of the protein. Phe tag, polyphenylalanine tag. The matrix is made up of phenylcephalose and it is eluted in presence of ethyl glycol. It is used for purification. Then there are S tag, S1 tag and S tag, which can be, the matrix used is monoclonal antibodies, and it can be eluted in presence of low pH buffer. Again, these are used for detection and purification. And the last is polyaspartate tag, and this can be, the matrix is anion exchange resin and can be eluted in presence of salt gradient. This is often used for purification. Host cells. So now protein expression vector need to find a suitable host for expressing the plasmid and expressing our gene of interest, because not all the cells can be a host cell, because for expression vector to produce or to transcribe a protein, to transcribe our gene of interest, it requires, depending upon the promoter that will be used, for example, in case of PET vector, we have a T7 lag promoter, and that would require a T7 RNA polymerase gene to be expressed also, and that T7 RNA polymerase gene should be tightly under the control of certain promoter, so that the expression is also not leaky. So in that case, the BL21D3 cells, which are used for the expression of PET vector, has a chromosomal copy that codes for T7 RNA polymerase, and which is under the control of lag operator. PET vector is most commonly used bacterial expression system. There are several others also. Just for the sake of simplicity, I am using PET vector as an example here. So PET as an expression vector contains following characteristics. Like it has an antibiotic resistant gene, that is ampicillin in this case, has a bacterial origin of replication, has a F1 origin of replication as well. It has a lac-I repressor gene, which allows a tight regulation of expression. It has T7 transcription promoter and multiple cloning site. Recombinant protein expression methodology. So now our gene of interest is already cloned into expression vector. How do we produce or express our gene of interest? As I mentioned in the previous slide, the vector need to be transformed into a host cell which contains the desired genes for expression of the protein. In presence of a lag promoter, we will add IPTG or lactose to induce the expression of T7 RNA polymerase that is present in the host genome, by which will remove the lag repressor. Same time, the same IPTG will allow the recruitment of T7 RNA polymerase on to the lag promoter, which is T7 lag promoter of our gene of interest, and eventually what will happen that we'll have transcription and translation of our gene and protein will be produced. The expression again explained in this schematic, as I mentioned earlier, that host cell contain a T7 RNA polymerase gene which is present in the genome as is shown here, but that is under the control of a lac promoter. So once we add IPTG, the lac-I repressor is removed, and E. coli, the host RNA polymerase, will synthesize T7 RNA polymerase. Now this T7 RNA polymerase can go and bind to the T7 lac promoter of the expression vector. However, the lac-I, and the meantime, the lac-I repressor, which is also blocking the T7 lac promoter, will be removed as the IPTG is added. So our E. coli RNA polymerase synthesizes T7 RNA polymerase, and the T7 RNA polymerase will go and start transcription of our target gene. Subsequently, our gene will be transcribed and translated, and protein will be produced. As in bacteria, transcription and translation is coupled. This reaction is very quick and will have a rapid production of protein. So here is an overview of recombination protein purification. Now we have expressed our protein into BL20D3. So we have got a lot of protein produced inside the cell. Now it is time to isolate that protein from there. So there are specific steps that are required to isolate that protein from inside the cell. First step is to lyse the cell, and cell lysis is depending upon the matrix you are using, as well as depending upon the expression of your protein. If a protein is expressed in an inclusion body, then you need to use a denaturing condition to for isolation. If your protein is expressed in a soluble form, you might not require a denaturing condition. But first step is to lyse the cells and get the protein out. Second step is to use the short-peptide tag, which we've been discussed in a previous slide. Based upon the short-tag peptide that we have in our protein, we use the matrix, and then which will be used to purify this protein from the mixture of cell lysate. And in the last step, we'll use the tag specific elutant to get our pure protein purified. Here is the schematic of recombinant protein purification methodology. So as I mentioned in previous slide, that as the protein is, our gene of interest is cloned into expression vector, transformed, and protein is expressed. Now we'll take that culture and we'll pellet cells, lyse them in presence of detergent, heat, by heating, by mechanical shearing, or by urea, so that our protein is burst out of the cell, or so the protein is released from the cell. Now this protein can be in a soluble form or in a form of a pellet. For the sake of simplicity, in this case we will discuss about a protein which is expressed in soluble form. So protein is expressed in a soluble form, is lysed, and now pellet is removed. The protein is in the supernatant and will be loaded into the column which contain the matrix, which will be specific to the tag which is present in the expression vector. In this case, suppose there is a histidine tag which is attached to our recombinant protein. The column will be of nickel and Ta. Once the protein is bound, non-specific protein is removed by adding washing buffer, and eventually the protein is eluted in presence of our elutant such as imidazole, and purified protein can be collected. So students, let's now summarize what we have learned in this module. So recombinant proteins are produced using expression vectors, and recombinant proteins are gene of our interest which we have cloned into an expression vector. Vectors are usually, these expression vectors are often plasmid, are of bacterial origin. An expression vector contains all classical features of a cloning vector, such as it has an origin of replication, it has antibiotic resistance gene, it has a primer binding site, it also has a multiple cloning site which allows us to clone our gene of interest or subclone our gene of interest from a cloning vector into an expression vector. Apart from a cloning vector, it also contains two other features. One is promoter, another one is short-peptide tag. Promoter is the region which allows the transcription of our gene of interest, and promoter sequences are, we have a range of promoter sequences that are commercially available depending upon our applications and the usage. What promoter allows us to have a control expression of our gene. So suppose our gene of interest is under the control of T7 lac promoter, as we mentioned, as it is common in PET series of vector. By using T7 RNA polymerase from the host cells, we can express our gene of interest into in a host cell. And the second is short tag peptides. These are the small DNA sequence which encodes for small peptides, and these peptides can be added at the N terminal or the C terminal of our protein. These are required for two specific purposes. One is to facilitate downstream purification process, and second, it can also be used for cellular localization as well as pull down of protein DNA complex or DNA protein protein complex. Thank you.